Hi there. Today's video is on osmosis. It's going to continue our understanding of how molecules pass through a semi-permeable, or also known as a selectively permeable, membrane. We just finished learning about diffusion, which is molecules moving from a high concentration to a low concentration. Osmosis is very similar. It's going to be the movement of water molecules from a high concentration to a low concentration. As you can see in our picture, we have a nice little drop of water to get you the hint that we're talking about water, and then we have a diagram with water molecules, a semi-permeable membrane, and some glucose molecules. Let's begin with our objectives. First and foremost, you're going to be able to describe the process of osmosis, what it is and how it works. Then you're going to be able to differentiate between the special types and processes and how we describe the solutions involved in osmosis: hypertonic, hypotonic, isotonic, and predict how a cell will react once it's been placed in one of these different solutions. Let's begin with the definition of osmosis. Osmosis is the movement of water from a high concentration to a low concentration until it reaches equilibrium. Just like diffusion, something is moving from a high concentration to a low concentration. In this case, we're just going to be watching the water molecules. This is a form of passive transport because no energy is required for this process to take place. Just like diffusion is also passive transport. So when we're looking at these situations, we're going to really be looking at the concentration of the solute on either side of our semi-permeable membranes, and that's going to determine the net direction in which water moves. So let's look at our diagram over on the right. This diagram is showing a container that has a semi-permeable membrane separating two different concentrations of solutions. Therefore, we have a concentration gradient. The selectively permeable membrane is going to let some things pass, but not others. So in this case, we want to look at the different concentrations on the sides of the selectively permeable membrane. On one side, we have some green dots representing sugar and some blue dots representing water. So here we have some solute and some water. On this side, however, we just have water molecules. So obviously we have a higher concentration of water molecules on the left-hand side than we do on the right-hand side. So what's going to happen? Water's going to move from a high concentration on the left-hand side to the right-hand side where there's lesser of a concentration. And that's simply what water is going to do when it's involved in osmosis and a selectively permeable membrane. It's going to move from a high concentration to a low concentration. Let's look at again a couple of examples. First one here is a diagram we saw in diffusion. In this, we have the red dots are representing solutes. On the left-hand side, we have lots of red dots, so we have a high concentration of solutes. On the right-hand side, not so many red dots, so we have a low concentration of solutes. In diffusion's case, we would expect the solutes to move from the left to the right. But now we're studying osmosis, so we're really interested in the water. So if we look at these, I'm going to be looking more at the background. So if we look at the side where there's not as many solutes, there's a lot of space for the water molecules to be. If you look at the side where there is lots of solutes, there's not a lot of space for a lot of water. So in that case, we would call this solution a high concentration of solutes, low concentration of water. Whereas on this side, the right-hand side, we have lots of water and low solutes. So the right-hand side has a higher concentration of water than the left-hand side. So we'll label those as high water content, low water content. And therefore we know the water is going to move from the high to the low, which is the right to the left. Let's look at an example of a cell. In this environment, we have a cell that's made up of 0.2 molar glucose solution and 0.2 molar sucrose solution. So overall, that means it has a 0.04 solute concentration. The environment I'm putting it in has 0.1 molar glucose solution and 0.1 molar sucrose solution. Therefore, this total environment has 0.02 molar solute solution. So I have to say to myself, all right, where do I have the smaller amount of solute? And that would be in the outside environment. So therefore, I have a higher water content. And on the inside of the cell, I have more solute, so that means I have less water. So I have a low water content. So which way is the water going to move? Again, water always moves from a high concentration to the low concentration. So it's going to move into the cell. Last very quick example down here. I have a 0.2 molar solution in the little bag and a 0.8 molar solution in the outside environment. So which one has the higher water content? Well, you're looking again at the solute content. The inside the bag has a lower solute concentration, so it has a higher water concentration. So therefore, the water is going to move out of the little bag and into the environment. So let's kind of start to put some words to these scenarios. So our first word that we're going to use to describe water situations is a hypotonic solution. And a hypotonic solution is one where you have low solid and high water. How do I remember that? Hypo reminds me of a hippo. Hippos tend to live in water, and they're round and they're fat, so they got to have a lot of water. So a hypo, high water, low solute. Hypertonic, on the other hand, has a high solute and a low water. I think of hyper. If you're a hyper little child running around, it means you probably just ate all your Halloween candy and you got a lot of sugar, so you have a lot of solute in you. So you have a high solute, low water. If we look at the diagram over here, we can have a good picture of a hypertonic situation and a hypertonic situation. Here I have low solutes, not that many dots, which means there's more room for water, so I have a high concentration of water. So that's my hypertonic solution. Over here I have lots of dots, so there's not so much room for water, so that's a hypertonic solution. Just so you know, generally most of our cells tend to be hypertonic. The third and final type of solution we want to describe is isotonic. And that one's the easiest of the three. It means you have equal amount of water with the solute. So it's easy. So let's get a little practice here. So we're going to picture these squares are going to represent a beaker with a cell inside of it. So if I look at the first one, I have not so many solutes, so that means I have a lot of water. We said, remember, a hippo is hypo, so we have a lot of water out here, and if we compare it to the cell, we have a lot of solutes, so there's not a lot of room. So in this sense, I have a hypertonic solution on the outside, the hypertonic inside the cell. So that means the water is going to move in. If I go down to this diagram, I have lots of solutes out here. That means I don't have a lot of water. I have lots of solutes inside. That means I don't have a lot of water. So this is going to be an example of isotonic. So where do the water molecules move? They're going to move in both directions. Some are going to move in and some are going to move out. There's not going to be a big net change. Last diagram, lots of solutes here on the outside environment. So that means there's not a lot of water. But if I look at the inside of my cell, I don't have a lot of solutes. There's very few dots, so that means there's a lot of room for water. So that means there's more water on the inside than there is on the outside. So that means water's going to move from a high concentration to a low concentration. It's going to move from the inside out. So what about if we give some examples of living organisms? The first living organism we're going to talk about is animals, and then we're going to use as a reference a protista. So protists are little tiny organisms that live in fresh water. And there's many organisms that live in fresh water. Think of fish. They're generally, we said, all cells tend to be hypertonic. So if they live in a hypotonic situation, water's going to be constantly flooding into them. If that's the case, they would always be filling up with water and bursting because a cell membrane can't hold an endless amount of water. It would rupture. So what do they have to do? They have to regulate their water. How do they do that? They have a very special vacuole. And that's this little one up here. If you look at this picture, it's called a contractile vacuole. A contractile vacuole is going to be constantly pumping out water so that it doesn't overflow with water and rupture and burst, or it will constantly pump out some of the solutes so that it stays a little bit closer to the environment that it exists in. So most animal cells are consistent of hypertonic in themselves, and they live in a hypotonic. So they have to have some kind of mechanism that they can remove the water by pumping it out. And one of those mechanisms is a contractile vacuole. What about plants then? How do they maintain the appropriate amount of water? What's best type of environment for them? Do they do better in a hypotonic environment, hypertonic environment? Why do you think that is? Well, let's take a picture. Look at these pictures, up at the top. If if I put a plant in a hypotonic solution, that means water is going to rush in, and it's going to fill the vacuole that exists. Remember, plants have a cell wall and a cell membrane. So all this water is filling in, putting pressure on the cytoplasm, which is going to put pressure on the cell wall. Because plants have a cell wall, it's going to hold that water in, create a certain amount of pressure that exists between that vacuole and the cytoplasm and the cell wall. We're going to call that turgor pressure. Plants like to have a lot of turgor pressure. It allows them to stand up firm and tall. Versus if I put a plant in an isotonic solution, water's going to be moving in and moving out. And then we're going to call that flaccid, and that's when a plant is slightly wilted. But if I put plants in a hypertonic solution, that means all the water is going to rush out. All the cytoplasm is going to pull away from the cell wall, and the cell membrane is going to shrink, and that's what we call plasmacized or plasmized. And it leads to a cell being shrinking and eventually dying. So not a good situation for the plant. So most plants, if we were to put what's the best scenario for them, they would prefer a hypertonic environment. It's going to build up their turgor pressure and allow them to stay firm and stand up tall. Otherwise, if they're in a hypertonic solution, they end up being plasmacized. Water's going to leave the cell, it's going to shrivel up, and it's going to die. So let's give a little bit of a summary here. Animal cells in isotonic are very happy and they're normal. Water's going to move in, water's going to move out, they're going to maintain their homeostasis. In a plant cell, however, that would be considered flaccid. They've got water moving in, water moving out. It's not as happy. Hypotonic in an animal cell, not a great situation. All the water's going to move in, the cell membrane can't hold it, it's going to fill and fill and fill, and then it's going to rupture. Plant cell, however, likes a hypotonic situation. It's going to build up its turgor pressure, allowing that plant to stand tall and be firm, and would be a happy little camper. Hypertonic animal cells, not a good situation. They're going to lose their water, shrink up, shrivel up, and die. Plants don't like hypertonic either. They're going to lose all their water, they're going to become shriveled and plasmatized and die. So basically, for living organisms, a hypertonic situation is not a good scenario. So let's get these in your notes. Cells placed in a hypotonic solution, the water's going to move in, animal cells will burst, and that's the term we're going to use for that is another word would be lyse. Remember lysosomes from your organelles, they break things down. Well, when we talk about animal cells swelling up and bursting, it's becoming lysed. Plant cells become firm, and we call those turgor, or they have turgor pressure. Put cells in a hypertonic solution, water's going to move out. In an animal cell, it's going to cause it to shrink or shrivel up. In a plant cell, it's going to be called plasmacized. And again, both situations, they're not good for living organisms. Isotonic solution, however, if I put an animal cell in it or a plant cell in it, there's a slight difference, but water will move in and out at an equal rate. Animal cells will become normal. Plant cells will become flaccid and wilty. And this is why we want to take a good peek at our plants and make sure that they have an appropriate amount of water. And that's what generally in a supermarket, that's why one of the reasons they continually spray our lettuces with water is so that they stay nice and full of turgor pressure, because no one wants to buy a piece of or a package of lettuce that is all wilty and it isn't appealing. So that about does it for our osmosis video. Make sure you know the definition of osmosis and passive transport. Make sure you know what a hypertonic solution is and you know the words turgor and lysed. Understand what hypertonic is and what plasmacized is, what is an isotonic situation and what is flaccid, and make sure you do a little bit of practicing determining which way the water will move in cells. I've added in the next slide a little bit of a link, a website you could go to so you can get some practice and some other pictures to help you have a better understanding of osmosis. Anything you don't understand, make sure you write it down and let me know in class tomorrow. See you then.